This is part 5 of a series of lectures on antimicrobial resistance and antimicrobial stewardship in veterinary medicine. In this part we'll be discussing antimicrobial resistance, where it comes from and how it arises and how it spreads. Among the things we need to talk about here are why antimicrobial resistance is an issue in the first place, how bacteria resist the effects of antimicrobial drugs and therefore become resistant, what the source of resistance genes are that confer this capacity to resist the effects of antimicrobial drugs on bacteria, how these resistance genes might spread between different bacteria. We then need to talk about what multiple resistance is and why it's important, and how it develops, and finally what we might be able to do to control this development of multiple resistance and how effective these controls might be. So the principal reason that we're worried about antimicrobial resistance is really conveyed in this slide. This slide shows the rate of antimicrobial discovery since the first antimicrobial drugs were developed back in 1936, and these were the sulfonamides. You can see that there was really a golden age of antimicrobial drug discovery where different classes of antimicrobials, starting with the penicillins, then the tetracyclines, chloramphenicol, the aminoglycosides, the macrolides, the glycopeptides and the streptogramins, and then finally the quinolones, were discovered in a period of just over 20 years. Then since 1962, really only 2 new classes of antimicrobial drugs have been discovered. So in the period of the last 50 or 60 years, we've really only discovered 2 new classes of antimicrobial drugs. And this really suggests that our options for new antimicrobials are starting to run out, and that one of the things we need to do is to take very good care of the drugs that we currently have and ensure that they continue to be successful into the future, because the likelihood is that we are not going to get a large number of additional classes of drugs in the future. Add to this the likelihood that any new class of antimicrobial drug is very likely to be restricted to medical rather than be allowed for veterinary use, and we see that we have a particular problem in veterinary medicine. So one of the concerns that arises when we start talking about antimicrobial resistance is who is responsible for its development. There is a common perception that antimicrobial resistance in human pathogens originates from animals. Now, while this is true for some of the resistance that we see in medical pathogens, much of the resistance that is seen in the pathogens that affect us as humans has its origins almost purely in medical use of antimicrobial drugs. However, ultimately, controlling this problem depends on understanding how resistance arises and how it spreads between bacteria and from one animal species to another. And these are complex issues and they have complex origins. And like most complex problems, an integrated approach is likely to be needed for appropriate control. So what is antimicrobial resistance? Essentially, it is reduced susceptibility to therapeutic drugs at the concentrations that we can achieve during therapy. This resistance can be innate or intrinsic to the bacterium that we're talking about, or it can be acquired. Innate or intrinsic resistance is resistance that bacterial species has always had, often as a consequence of its particular metabolic pathways. Acquired resistance is typically resistance that has been selected for or has been transferred into that bacterium from externally, and it is a relatively recent acquisition of that bacterium during its evolution. This resistance may be transferable between different bacterial species, or it may be resistance that is only inherited within that particular bacterium and its progeny. The major concern we have when we're talking about antimicrobial resistance is acquired resistance, and predominantly we're concerned when it is transferable, because this is the resistance that can ultimately spread to a range of pathogens and can result in multiple resistance. And as I have mentioned in a previous lecture, it's important to remember that antimicrobial resistance is not just against drugs. There can also be resistance against disinfectants and against heavy metal ions as well. So intrinsic resistance occurs when some bacteria lack a target for or are impermeable to particular antibiotics. And this means that this antibiotic or these antibiotics will never have an effect on any member of this bacterial species. In contrast, acquired resistance occurs when bacteria that are normally susceptible to a drug become resistant to that drug through either acquisition of a mutation or through acquisition of a completely new gene. Transferability occurs when some resistance genes are spread from one bacterium to another horizontally. And multiple resistance occurs when these resistance genes are accumulated by particular bacteria in the one organism, generating a multiply resistant organism. And this is the major concern in development of antimicrobial resistance. This multiple resistance can also include resistance to disinfectants and to heavy metal ions as well. So the consequences of the development of multiple resistance, or resistance in general, is that we have reduced efficacy of antimicrobials across a range of pathogens that are of importance in both human and animal health. This results in limitations on veterinary treatment, so we have fewer drugs that are able to effectively treat diseases that we see. It results in limitations on the efficacy of medical treatment, again because there are pathogens that are no longer able to be treated as readily as they could be before. It results in food safety and environmental concerns. It results in concerns about owning animals and about the risks that they might pose to their owner's health. For veterinarians, it particularly can have occupational health and safety concerns that can be raised about employees in the workplace. And one of the real problems with antimicrobial resistance is that if we wait for the ultimate proof that a particular practice is responsible for the development of resistance, it will then be too late for us to act at all. So if we're considering how we might approach control of antimicrobial resistance, we can consider a number of potential critical control points in the development of resistance that we might want to impose some sort of restrictions on. We may look at where these resistance genes come from in the first place and whether we can control the source of these genes. We might want to look at how these resistance genes spread between bacteria and between animals and see if we can prevent that. We may want to stop the development of multiple resistance, so this accumulation of different resistance genes in the one organism. We might want to look at whether we can control the spread of these resistant bacteria. We might want to look at whether we can control the selection of different resistant bacteria. We might want to aim our controls at the reservoirs of these resistant bacteria, or we may want to impose our controls on infection and on where these resistant bacteria actually infect animals. Now, ultimately, we're probably looking at multiple critical control points rather than a single critical control point if we're trying to prevent this as a problem throughout the wider community. So how does antimicrobial resistance develop? There are a number of different points at which bacteria can develop a mechanism for preventing the effect of an antimicrobial on their growth or survival. And these include the development of degradative enzymes that break down the drug into components that are no longer active. They can include efflux pumps that pump the drug rapidly out of the cell, the bacterial cell, and outside of the bacterium. It can result from an increased production of the particular target molecule for that drug so that there is more capacity for the bacterium to continue to function because it has more of that target protein sitting in its cytoplasm. There can be some sort of modification to the target of the bacterial drug so that it can no longer bind to that target structure in the bacterial cell and so no longer have an effect. And finally, there can be modifying enzymes within the bacterial cell that alter the structure of the drug so that it can no longer have an effect inside the cell. So if we start thinking about these different critical control points, the first of these is where these resistance genes arise from in the first place. So they can arise as a result of acquisition of random mutations in chromosomal genes that ultimately result in decreased susceptibility of that particular organism to the drug. This type of resistance occurs quite commonly, but is less commonly transferable to other bacteria. Alternatively, bacteria can acquire a gene that confers resistance upon it from other organisms. Where these genes have come from has often been considered a bit of a puzzle. They can come from antibiotic-producing organisms. They may come from commensals and other pathogens that occur in the gastrointestinal tract or on the skin of animals, or they may come from environmental organisms. What is clear is that these acquired genes are much more likely to be transferable to other bacteria than genes that have acquired a random mutation and that sit on the chromosome of the organism. Most of the antimicrobials that we have available to us for therapy have their origins in environmental bacteria and fungi. These antibiotic-producing bacteria and fungi all contain resistance genes that enable them to survive the effects of the chemical that they're producing themselves. These environmental bacteria commonly survive in a habitat where antibiotics occur, and they are encountering these all the time. Some of the resistance genes that we are interested in protect bacteria not only from these particular drugs, but generally from environmental toxins. What this has meant is that there is a very large pool of potential resistance genes in environmental bacteria. And some environmental bacteria can even live on antibiotics. They can use them as a primary energy source. Recent studies have shown that resistance genes can be found in 30,000-year-old soil samples from the Arctic tundra, making it clear that antimicrobial resistance clearly predates our use of antibiotics as a therapeutic option. And most of the transferable resistance in pathogens is likely to have originated from environmental bacteria. What this all means is that we are unlikely to be able to control the source of resistance genes because they are in the environment of our animals all the time. The second critical control point we may consider is the mechanisms of spread of resistance genes between bacteria. Many of these resistance genes are carried on mobile DNA elements such as plasmids, transposons and integrons. Even resistance genes that aren't on these transferable elements can sometimes be transferred by some of the mechanisms bacteria have for sharing their pieces of DNA with each other. This transfer is most commonly seen between close relatives, but it has been seen between quite distantly related bacterial species, and can even be promoted by the exposure to antimicrobial drugs. This means that it is quite difficult to control the spread of resistance genes between bacteria. The third point we may wish to control the development of resistance is the development of multiple resistance. Multiple resistance occurs because mobile genetic elements can be accumulated by bacterial cells. So as multiple mobile elements are accumulated by the same bacterial cell, we end up with clusters of resistance genes and other beneficial genes for that bacterium on the chromosome or in plasmids or integrons in that organism. And integrons in particular appear to be able to capture multiple resistance genes at the same site on a bacterial chromosome. Once these clusters of genes have been developed, they can then be transferred as a single block. And so what we have is transfer of not only single resistance, but transfer of multiple resistance between bacteria. This development of multiple resistance is likely to be promoted by environments where multiple antimicrobials are used simultaneously or in close succession. Now here we do have an element of antimicrobial therapy that we are likely to be able to control, and that is the use of multiple antimicrobials at the same time or in close succession. So the next point at which we might consider controlling the spread of antimicrobial resistance is the spread of resistant bacteria. Although antimicrobial resistance genes can potentially travel around as pieces of naked DNA and be transferred just as a piece of DNA, More typically, resistance genes are dispersed by sitting within a bacterial cell. And so the mechanisms that we typically associate with the spread of infectious organisms, such as physical forces like wind and water, and the movement of contaminated soil, food and water, the movement of animals, can all be associated with the spread of resistant bacteria from one place to another. What this means is that the sorts of measures that we typically put in place for biosecurity around our domestic animals are methods that can also help control the spread of resistant bacteria. Unfortunately, one of the most potent mechanisms of transfer of resistant bacteria is the movement of people, and this is the one point where we have the least control over biosecurity. Resistant human pathogens have often had the most rapid and the greatest dispersal around the globe, and this is probably the area where we have least capacity to influence the spread of antimicrobial resistance. Be that as it may, we can still ensure that adequate biosecurity is imposed on our domestic animals and focus not only on some of the infectious diseases that we are more commonly focused on, but also on the dissemination of resistant bacteria. So the next point at which we may wish to impose some control on the development of antimicrobial resistance and on its dissemination is on the selection of resistant bacteria. Exposure to antimicrobial drugs provides a selective advantage to those bacteria that are resistant to their effects. Therefore, treatment with an antimicrobial drug allows resistant strains to increase in number and also potentially enhances their capacity to transfer resistance to non-resistant strains of that same organism. Multiple resistant strains can be selected by any one of the antimicrobials that they are resistant to, and this co-selection of antimicrobial resistance gene cassettes can greatly enhance their spread. The other thing we need to consider is that antimicrobial use can have a prolonged effect on a bacterial population because it disturbs the balance in that population. This graphic depicts the mechanisms that can contribute to the development of resistance and particularly the development of multiple resistance by bacterial cells. There are multiple mechanisms that can be used by bacteria to acquire pieces of DNA from other bacterial cells. These mechanisms include transduction, transformation, conjugation and transposition. All of these mechanisms ultimately can result in the development of either chromosomes or plasmids that contain a large number of different antimicrobial resistance genes in the same place in the bacterial cell. Once these collections of resistance genes have all been accumulated in the one place, every time any one of the drugs that bacterium is resistant to is used, you get selection for all of the resistance genes at the same time, and you can very rapidly get dissemination of this collection of resistance genes throughout different bacterial populations. The last point that we may wish to impose some controls on the development of antimicrobial resistance in our domestic animals is on the reservoirs of these resistant bacteria. Unfortunately, resistant bacteria can reside in a number of different places. They can be environmental bacteria, they can be commensals, they can be pathogens that we really are concerned about in the development of animal disease. They can be sitting in wildlife and in feral animals. In mixed farming systems, they may be residing in other animal species other than the species that we're primarily farming, or they can be sitting in humans. One of the biggest problems we have is that carriage of resistance genes often does not sufficiently diminish the fitness of the bacterium containing that resistance gene enough for there to be rapid selection against resistance. And so as a result, once those resistant genes have been required by a bacterial cell, they tend not to be lost for a very long period of time.